In your email, there's a link in your email for you to assess me. Please don't just fill out the multiple choice ones, type out in comments. Okay, let's see here. So I believe I finished week 10 and 11 and starting on week 12. Is that right? That sound right to you guys? I defined antiseptic and disinfectant. I think that was the last thing.
Yes.
So we're on week 12 here. Understand that ultraviolet. Oh, sorry. So finished antiseptic and disinfectant right there. And 12. Understand that ultraviolet light may inhibit microbial growth. That's all you need to know. We saw some evidence of that with the experiment. The longer that microbes are exposed to ultraviolet light, the more, I don't have on this study guide, but you need to know it, what is produced. The more the DNA is exposed to ultraviolet light, the more you have the production of what? Thymine dimers. Ultraviolet light produces thymine dimers, which is damage, not good. Know the name, function, and significance of all the components of the plasmid transformation experiment. What is a plasmid? What is bacterial transformation, etc. So basically the terms from the quiz. Quiz information. Do I need to go over any of that stuff?
Sure.
You need to know the name of the plasmid, P glow, lowercase p, and then all caps, GLO, P glow. And then, I wrote it up on the board with the circle, with ARA, GFP, and bar. You have that in your notes. And then just the basic steps of the transformation process. That's just quiz information. Do I need to go over any of that?
You just need to,
The first
you just need to know how to write the circle of that, or how are you going to ask?
Well, I can't tell you how I'm going to ask you, but you're not going to need to draw all that, but you just need to know what it's made of. Just the nature and the structure of the plasmid. It has those genes on it, the function of those genes, the ARA is required for transformation, the GFP is the green fluorescent protein, which fluoresces under ultraviolet light. It's all the quiz information. Wherever you wrote that down. Same stuff. Nothing new.
Are you recording?
Week 13 and 14. Know the pH indicator in Snyder auger. That's on the table. Remember, you're active studying. Don't just look automatically. Try to think of it. And that's listed right here. Here's Snyder. Broad Pressel green is the pH indicator. Know what microbes are tested for. This is quiz information. Lactobacilli, streptococci. The names of the white blood cells. Remember, that's all you needed to know, just the names. Never let monkeys eat bananas. Neutrophils, lymphocytes, monocytes, eosinophils, basophils. You just need to know the names of them. I'll be mentioning them again today. Probably need to know a little more information for lectures. The ELISA test, again, just the quiz information. This is a little redundant here what I have, so it's just the quiz information. What is the purpose of it? Know each step, the name and purpose of each component in the test. These are a little bit redundant asking the way to ask this. As long as you know all the quiz information for the ELISA, you're good. The steps, the 4 steps. Any questions on the ELISA? Okay, let me go through the PowerPoint briefly. So this is also, you need to study the PowerPoint, you're gonna study that, the study guide, the table, and the PowerPoint. I could have these pictures from the PowerPoint on the test. You're not gonna have real cultures on the test, just to avoid spilling.
Pen or pencil? Okay.
That you'll have pictures. So you should go through this, memorize the pictures, memorize all the information on it. The phenol red sugar fermentation, remember the PRS? We had different types, and then it just gives tells you what's positive and what's negative here. The right is positive, the left is negative. I mentioned the Durham tube for gas production. So it's basically just going through all of this. I'm not going to read through all of them word for word, because it's all there for you. The indole test, you should know that it comes from the SIM medium. This is the SIM medium. You used it for the unknown as well. Testing for indole. You added a reagent to that. Active studying. Think about what that reagent's called. Does anybody remember? By any chance? What you added to test for indole? Kovacs, right. If you can't remember it, go to the table. It's listed on the table. If it's a positive, it produces this red band. Some of you, for testing for your unknown, you got that. Some, it was negative. On the left, it's negative. The methyl red test. On the right, you have to add a reagent for this. Do you remember the name of the reagent for this? Methyl red. That's easy. Which happens to be a pH indicator, and it's the reagent because you added it. Positive on the right, negative on the left. And I think I mentioned to you, you're testing for mixed acid, and that's due to the fermentation of glucose in this medium. But you're testing for mixed acids. Voges-Proskauer, you're testing for neutral end products, which I also told you in lab. Neutral end products. Do you remember the reagent for this one? It starts with a B. Barrett's. Barrett's A and B. Barrett's A and B. It's not Voges-Proskauer A and B. The bottles used were, it said VPA and VPB. But the actual reagent that you need to know is Barrett's, which is on the table. Positive on the right, negative in the middle, and then uninoculated on the left. Voges-Proskauer. If you remember, this is where you split 1 mil and 1 mil from the MRBV. MRBV. Citrate utilization, this is pretty straightforward. Blue is positive, green is negative for citrate utilization. Pretty straightforward. The lysine decarboxylase, purple is positive, yellow is negative, and then there's some other information here you need to know. This is pretty straightforward. DNase, if there's clearing around the growth, it's positive for the enzyme and for the destruction of DNA. DNA has been put into this plate. Pretty straightforward. Urea, positive is this fuchsia color, and this is negative or uninoculated for the breakdown of urea or the presence of the enzyme urease. Do you remember the pH indicator in this? Did I tell you that? It's phenol red. I think I have that for you. Yeah, phenol red. And what pH causes that reaction, that color? Alkaline or acidic? Alkaline. That's phenol red. Acidic is yellow. Gelatin hydrolysis. Gelatin gets liquefied by some microbes, as indicated in B. It stays solid if it's not hydrolyzed. If they have the enzymes to do it, it liquefies. Again, I want you to know, and you needed to know this for the first lab exam, why we use agar over gelatin for solidifying agent, which is right here. Most bacteria cannot digest agar. Many more can digest gelatin. And agar stays solid well above room temperature. You need to know that for the first exam as well. But now we actually use gelatin. Because gelatin is a solid, like jello. Same thing. Except microbes can break it down. You don't want it, your Petri plate getting all liquefied in the middle of incubation. That'd be pretty messy. Phenylalanine deaminase, this is straightforward. You have to add a reagent for this one as well. If you can't remember, you go to the table, it's ferric chloride. It turns green if it's positive. It does not if it's negative. Catalase production, bubbles are produced. It's sort of hard to see them, but there's bubbles here. You didn't do it this way for the catalase test. You took a slide, you took a swab, took some bacteria out of your tube, smeared it onto the slide, on the same slide, a little bit there, a little bit there, and then you added what? Hydrogen peroxide, right? And you put Staph aureus and Staph epidermidis on it. And Staph aureus was the one that was positive. You don't need to know that for the test, which microbes, but just refreshing your memory. So just remember that when you're looking at this, you didn't do it with the tube, you did it with the slide. But the same concept, bubbles. Bubbles is the oxygen produced from the breakdown of H2O2. Then your blood agar. We're looking at hemolysis. Gamma is no red blood cell destruction. I put here, is blood agar selective, differential, or enriched? If it's just plain old blood agar, not Columbia CNA, which is in the first exam, but you don't need to know that one. But just blood agar in general is enriched and differential. It's only selective if something special is added to it. The Columbia CNA had something added to it to make it selective. But blood agar, just regular blood agar is differential and enriched. Differential means it differentiates something. What's it differentiating? Hemolytic properties, hemolysis, alpha, beta, gamma. And it's enriched, which means it can grow fastidious microorganisms, the picky eaters. So this is gamma because, as you see on the plate, there's really no discoloration of the medium. Whereas alpha, partial red blood cell destruction, you can see it's a greenish color here. Partial red blood cell destruction, the growth. And below the median, below the growth, the median below the growth is what's changing color here. Not too much like along the edges. This is all growth right here. This is bacterial growth, all of this. And then beta, complete destruction. You see a single colony there, the dark dot, and then around it is very light. That's where the red blood cells have been completely destroyed. Beta hemolysis. Okay? Kleigler's iron agar. I went over this in depth in lab. This is with the code. Do I need to go over this? Any questions about this? No? You have uninoculated, this tube here, it's yellow at the bottom. Remember, the four places, you have the slant, the butt, gas, H2S, just to refresh your memories. And then basically, if it's all red, which is not, the tube on the left is uninoculated. If it's all red, it's uninoculated, or there's no sugar fermentation, there's no fermentation if it's all red. What would the two places be? The letters. KK, which would be alpha. If it's all yellow, what are the two places? AA. If it's yellow on the bottom and red on top, Ka, glucose only fermentation, which I have written in here. And that's because glucose is present in a much lower amount. You can see gas production here. crack, the gaps, pushing up the agar, and then the black precipitate, H2S production. So, and what about fermentation in this tube? Yes. Because remember, you didn't need to know this for the test, but you need an acidic environment to make the H2S. So technically, it's yellow in the butt. So this is glucose only production. So this is Ka minus plus, which is how I tell you right here. Okay, so you have this in your notes. Any questions about that? Okay. The coagulase test, pretty straightforward. You inoculated the plasma medium, and if the microbe had coagulase, it solidified it, as you see at the top. The plasma medium was liquidy, and then it got solid or semi-solid for a positive result. The nitrate reduction. Now, this is the other one that was a little bit complicated. I'll go through this now. So, if you remember the equation in the lab book, you have nitrate. We're testing for whether nitrate is reduced. If you can picture the equation, you have nitrate is reduced to nitrite, and then the nitrite can be converted to some other compound, like 4 other compounds are listed, which would still be a positive. If the nitrate is converted to any one of those things, it's positive for nitrate reduction. What you tested for was whether there was nitrate present, nitrite present, or one of the other things. You weren't testing for one of the other things directly. Let me go through it. So, first you added reagents A and B. If it turned red, it was positive for nitrate reduction. It meant that nitrite was present. A and B essentially react with the nitrite. So E. coli there would be positive. These two, no color change, so we don't know. Either nitrate is still present, which would be a negative, or the nitrate was converted to one of those other compounds, past nitrite. How do we determine that? Remember how? What did you have to add? Zinc. You had to add zinc, right? And remember, zinc reduces what? Nitrate. Zinc reduces the nitrate. Okay? So, E. coli was positive because it turned red after adding A and B. These other two, we don't know yet. We have to add zinc. So after adding zinc, which is reagent C, Pseudomonas aeruginosa is positive, no color change. So there's no color change after adding zinc. It's positive. Why is it positive? Because if you added zinc and there was no color change, it meant there was no nitrate to be reduced. Zinc reduces nitrate. So there was no nitrate to be reduced. And there was no nitrite. We would have known if there was nitrite because it would have turned red already. It was one of those other compounds that was present. Whereas Corynebacterium xerosis, it's negative because there was a red color change. It turned red because zinc reduced the nitrate that was still present to nitrite, and the nitrite reacts with the A and B that you already added. And again, if it's still a little confusing, read the book. It's crystal clearly explained in the book. Crystal clearly, if I can say that. And also, yes, I mentioned this as well, I might have no more than 3 questions max on this stuff. So don't be spending 50% of your time trying to get this down. I'd sort of wait till the end to study it. It's only going to be a small percentage out of the total. Oxidase test, remember, we did not do this one because it never works, but you still need to know the name of the test and what a positive looks like. Purple. After you add the oxidase reagent, it turns purple. It doesn't turn purple, it's negative. So this is straightforward as well. Bile escalin, a brown coloration on the plate next to the growth, around the growth, indicates the escalin is degraded. And it means either group D streptococci or enterococci is present. These are two groups of bacteria. Group D streptococci, which includes certain strains of species of streptococcus, which you probably have not heard of. And enterococci. It's really the enterococcus that's usually more significant, Enterococcus faecalis. But both of these are tested for. They make the entero tube, I went over that with the code. I'll show you how to do that. And that's it. Any questions on the lab exam? So it's going to be Monday. I don't know what room it's going to be in yet, so just go up near our actual lab room. Oh, probably what we'll do is we're going to go into our lab room first because I want to show you the results from last week. Briefly, it'll take just a couple minutes, and then we'll go into wherever we're going to be testing. Hopefully, it's not going to be in that room, and it will be at. You're going to have to come to lecture, because I'm not sure when we're going to have it yet. So, and if you don't come to lecture for some reason, I would make sure you're up there at 10 o'clock, just in case I have it at 10 o'clock. And if it's not, hang around there, don't get lost. Walk all the way down to the very corner. We had it in the very corner of the back, right, last time, in case we're there. No excuses, because you know it's going to be up there somewhere around that time. Okay, any questions? Okay, lecture time. Any questions on this table? How to interpret this? Pretty straightforward. Again, so you got to memorize this. These are all the different tests in this column. The name of the medium and the test itself. You have positive results, what that looks like. You have the negative results, what that looks like, and the pH indicator reagents. You can always come see me during my office hours today or tomorrow if you have any questions. I don't have office hours Friday. Okay, so hopefully you read ahead of time, as I suggested that you do, especially for these two chapters. Probably more so for the next chapter. Non-specific defenses. So we have two chapters on immunology. Non-specific defenses as opposed to the first one, just the terminology, susceptibility, lack of resistance to disease. Everybody has a certain amount of susceptibility. Everybody gets sick. Some people more than others. Resistance, we have our resistances, our immune system helps fight off, and other defenses help fight off disease. Non-specific defenses or resistance, defenses against any pathogen. It's not specific. Like your skin is a defense. It's part of the non-specific defenses, which I'll talk about in a little bit of detail coming up. And then specific resistance, technically that's immunity, although I use the term immunity for both commonly. Resistance to a specific pathogen, that's next chapter. So we have non-specific resistance, and you have these defenses. The first line, skin, mucous membranes, your normal microbiota. I'll talk a little bit about each of these things. And then your second line defense, cells, actual cells. And there's going to be a little bit of specificity here, but the book calls it non-specific. There's some ways of specificity. Inflammation, fever, microbial substances. I'll touch on all this. And then this is not chapter 17. This is from the full edition of your textbook. We have the custom edition. Ours is chapter 14. So this is the next chapter. We're going to talk about B cells and T cells and antibodies. Next chapter. So let's start. So non-specific defenses, I mentioned the skin, mechanical factors, your book is characterizing it as. The skin is very formidable in terms of preventing microbes from getting in your body. You have the epidermis, tightly packed cells with an outer layer of dead cells with keratin, a protective protein. So your outer layer of cells, some of you might know this from A&P, they are dead. And they're continually sloughing off. They slough off, fall off, carrying with it potential pathogens. So they're always, all the microbes on your skin are continually falling off your body. So that's just, that's one way. And then the cells underneath, of course, are dividing and replacing it. It's dry. That's another part of the skin that makes it a defense. You need moisture, water, for microbes to grow. It's relatively dry. Of course, when you sweat, there's more moisture there, but most of the time it's relatively dry. There's oils and things. But I'm talking about water as far as dryness. Relatively dry. So it's not a great environment. If you go to a tropical area, like tropical, Florida and parts of Florida is subtropical. I'm talking about jungle, tropical. The first time I went to Africa, I was in Liberia, which is in the tropics. It's fully, rainforest. And it's suggested that you take a shower twice a day there. Because it's so humid, and your skin's always wet. And you can get an infection, skin infection like that. I don't know if I told you this story, if it's too much detail. I was in another country, Ghana, and which parts of it are rainforest. It's not quite bad as Liberia. But I spent, stop me if I told you the story. I've been to Africa several times in the backpack and stuff. And I carry 2 pairs of pants, one I'm wearing and one in the bag. And if I'm doing what I'm supposed to do, I wash the pants every day. Take them off, wash them for the other pair. Well, I went probably 3 days without washing them, sweating, stuff like that. I can't remember why. It was hard to get to be able to wash it. It's Africa. And I started getting a rash around my waistband, right there. And as soon as I felt the itchiness and looked and I saw the little red, I'm like, oh. And what I did is I got a place to stay with a fan, and I just stayed in basically the room for like 4 days straight without getting sweaty. But so I can wash and keep it clean and as dry as possible. But even doing that, as soon as you take a shower and you come out and dry off, you're sweating with it. You're starting to sweat already. I did get there was no air conditioning. So it was fan basically. And it's just that's the way it is. It just took. And I knew me. I knew how my skin reacts, and I'm very susceptible to skin infection. I get poison ivy just looking at it. So I knew I had to be very careful because I knew people that actually had to leave the country from skin infection because it just took off and they couldn't get a hold of it and they had to get out of there because it can kill you if you let it go, and it breaks the integrity of the skin and gets into the underlying tissue in the blood. It sounds like nothing, but it could be major stuff over there. So I just okay. And it took a few days, and finally, then after that, I washed it regularly. But we don't have to worry about it as much here. But of course, if you go days without washing your clothes, you could get skin infection as well. Picture of the skin, you have multiple layers of cells. So you have the epidermis, and then the outer layer with the keratin. You have the dead cells up in here. And you can see the individual cells. You can see the cell walls there. Look how small it is. So you got, you know, tons of cell layers there. You got a little bacterial cell at the top. There's no way it's going to get through that. The only way it gets through is in a break in the skin or through pores, for example, or hair, where the hair comes out. So it's pretty formidable, the skin. You've got mucous membranes, which is also a mechanical factor. In the respiratory tract, you've got the ciliary escalator. Moving back up. So mucous membranes, the lining the respiratory tract, lining the gastrointestinal tract, for example. Mucus. That's a barrier. It traps microbes. Not perfect, but it's a pretty good barrier. Microbes have a hard time getting through that to underlying tissue. So mucous membranes. Ciliary escalator, that's separate. Microbes trapped in mucus are transported away from the lungs. Everybody's got cilia in their windpipe, and you have mucus that's secreted. You breathe in, microbes gets trapped in the mucus, and then the little hairs are always beating, comes out. You know, clear your throat a little bit, swallow. That's, you know, somebody calculated that you swallow about a gallon a day. Saliva and, you know, the mucus that's coming up, and swallow it back down. Ciliary escalator. Smokers, probably mentioned this at some point in class. Smokers, your ciliary escalator slows down. And the more you smoke, the slower it's going to get. I don't know if it can actually stop or not. Ask your AP instructor. But smokers are more susceptible to infection, partly because of the cilia are being damaged. They're slowing down. You can't get out the mucus with the bacteria. The lacrimal apparatus makes tears. Washes the eye, so you're constantly making tears from the lacrimal apparatus. Keeping the eyes flushed out somewhat. Saliva washes off microbes, keeping it from attaching. Urine goes out, so microbes can attach. Vaginal secretions as well, flowing out, keeping microbes from attaching, causing potentially disease. So we got physical factors, physical barriers, essentially, are one part of the defense. Chemical factors. You got different substances that microbes make. Bacteriostatic and fungistatic, fatty acid, and sebum. Sebum is the oils that you secrete. You have oil glands, essentially, secreting sebum. In that, there's fatty acids that are bacteriostatic and fungistatic. They inhibit growth of these microbes. So you have dryness, for one thing, there's no water, it's an acidic environment, slowing down growth if not stopping growth. Low pH, going back to this, the low pH of the skin. Most microbes, of course, most bacteria want a pH 7. Did we do the pH lab? No, I don't think we did. We used to do that. I think this is the first semester we didn't do it. Inoculating a bunch of different tubes with pH 2 up to like 10. They can grow very slowly, many microbes at that pH, at 5, maybe not 3, but their optimal is mostly around 7. Fungus, a little bit lower, if you recall. Lysozyme, one of your defenses. You find it in pretty much all around your body. Perspiration, tears, saliva, tissue fluids, throughout your body, you have lysozyme. It's an enzyme. We saw this before. Do you remember where we saw this before? What? Right, the bacteriophage. The bacteriophage has its own lysozyme that it used to make a little hole in the bacterial cell wall, injecting its DNA. Remember that? And the cell lived, but then when it replicated a bunch of itself, and then when it to get out, there's thousands of these bacteria, these viruses, using the lysozyme, which lysates the cell. Well, you have the lysozyme also. Isn't that cool? A little virus that infects a bacterium has an enzyme that is very similar to an enzyme that you have. If that's not the unity of life, I don't know what is. So very, very similar enzymes. It does the same thing. It attacks bacterial cells indiscriminately. Not all bacteria are susceptible to it, but many are. So you got your tears, lysozyme all in there, saliva, tissue fluids, perspiration, you got everywhere. Obviously, not all bacteria are susceptible because we have bacteria living on us and we get sick with bacteria, so it's just one part of the whole system. Low pH in the stomach and gastric juice 1.2 to 3.0 roughly, so a lot of bacteria will be killed on the way through the stomach. They won't grow for sure, most of them in the stomach. And then another compound, transferins, in the blood bind iron. So you have these compounds called transferins that bind to blood, bind to iron. Microbes, remember, have siderophores, which also bind the iron for themselves, if you remember that term. So it's like a battle for the iron. The transferins bind it so they can't get it. And then your normal microbiota, I've touched on this before, that they are a defense against pathogens. They can be, using microbial antagonism / competitive exclusion. Normal microbiota compete with pathogens. Antagonism, competitive exclusion. So your own normal flora, remember that term, flora, same thing. They're competing with each other and potential external pathogens for nutrients. They make antimicrobials. Your normal flora make antimicrobial agents. I've touched on this before. You've got hundreds of different species of microbes in and on your body. They're fighting each other. They're fighting each other for nutrients, and just by that fact, they're excluding, competitive exclusion, potential pathogens from outside as well as within. They're making antimicrobials to kill each other. And anything coming in from outside usually can't handle it, because there's very few of them usually that try to get into the body. So it's a very powerful mechanism. And let me just go on a side thing, don't write this down, but we've shown that not only just this antagonism and competitive exclusion helps protect against getting sick, but it also, you can imagine this, it promotes your immune system as well, your microbes. They've shown this because they've grown sterile animals, completely sterile mice. So they didn't allow any microbes to grow in or on mice. And they showed that their immune systems were much weaker than animals that are allowed to be colonized. Natural birth from its mother and living out in the open and stuff like that. The immune systems were much different. Pretty interesting. All right, so the blood cells. That's where most of the immunity and the defenses play out. Red blood cells, of course, and then our white blood cells, which are called leukocytes. Those are the immune cells. You have the neutrophils, the basophils, the eosinophils, the monocytes, the leukocytes. So we're not concerned with the platelets. So these are the 5 different types. And the mnemonic never let monkeys eat bananas. The percentages, I said that mnemonic tells you the percentage from high to low. And you're going to need to know that. So the neutrophils are the most abundant white blood cell in the blood, 60 to 70% in a healthy person. Lymphocytes, the next most abundant, 20 to 25%. Monkeys, monocytes, 3 to 8%, eosinophils 2 to 4, basophils 25 to 1%. I do want you to know the rough number for the neutrophils and the lymphocytes. So know that it's around 60 to 70% for neutrophils and 20 to 25% for lymphocytes. The other ones, you don't need to memorize the numbers, but just know the order of abundance from high to low. Neutrophil down to basophils. Okay? It's in a healthy person. If somebody's got a bacterial infection, the numbers are going to be off. You'll probably have a higher neutrophil percentage, for example. Because they're fighting off a bacterial infection, so they're going to make a lot more neutrophils. Or if they've got HIV and they're not responding well to the drugs, their lymphocytes, T cells mainly, are going to drop. So, a little bit about them. Neutrophils are phagocytic, which means, as you know, they eat other cells. And there's a pretty cool little movie that was made in, I believe, in the 50s, showing chemotaxis, about that in a second, and phagocytosis. So these are all the red blood cells here. These are red blood cells. This is a neutrophil, and then the little dots are bacteria. And you can see the shape, coccus, with like diplococci, like 2 round cells stuck together. And it's chasing it, and then it's going to engulf it right there. And then we'll break it down. Chemotaxis, remember, is movement toward or away from a chemical. We'll talk more about that later in this chapter, but that's how it's chasing it. There's chemicals that are concentrated around the bacteria. That was sort of speeded up, but it was attracted to certain chemicals near the bacteria and was able to catch it that way. So that happens in your body when neutrophils are attacking microbes. So they're phagocytic, the neutrophils. Basophils produce something called histone. You don't need to write it down now because I'll show it again later. Histamine is something that dilates blood vessels. Vasodilator. It's coming up again, so you don't have to write it down now if you don't want to. Basophils produce histamine. Eosinophils, mainly involved in parasitic infection, like helminthic worms. Eosinophils are involved in that. There's some phagocytosis by eosinophils, but they're mainly involved in killing off parasitic worms. And we'll see an example of that coming up. Monocytes are phagocytic after they mature into something called macrophages. So the macrophages are phagocytic. Monocytes are in the blood, and then when there's an infection, they will differentiate in the macrophages and become phagocytic. Just a little more about macrophages. There's two different classes. So there's two classes of macrophages. There's fixed and then there's roaming. Fixed macrophages, they're pretty much everywhere in the body. Various tissues such as liver, lungs, nervous tissue, they're pretty much throughout your entire body. Fixed. Sort of just hanging out in different tissues, and if any bacteria come in, they'll try to eat it. They're not perfect, they can't always eat it, but they're pretty good. And then there's wandering that roam and actually move around throughout the body. The lymphocytes, that's the next chapter. Involved in specific immunity. The B cells and the T cells. Those are lymphocytes. E lymphocytes, T lymphocytes. Next chapter, we talk about that. This chapter is non-specific immunity. Okay. I talked a little bit about the lymphatic system when I mentioned the elephantiasis, the parasite that clogs up the lymphatic system, and many of you probably have studied the lymphatic system in A&P, but this is a good picture showing the lymphatic vessels. The blood vessels are not shown here, but they're like blood vessels in that they go throughout the entire body like this. And as I mentioned before, the blood vessels leak fluid normally. That fluid has to get back into the blood. The elephantiasis, remember, the lymphatic system got clogged with the parasite. So any fluid that leaves the blood and goes into the lymphatic system cannot go back into the blood. It's returned back into the blood somewhere up there. Some of you might know exactly where, I don't remember, but somewhere up there it goes back into the blood. But if it's clogged, that person's leg, remember, was swollen, the left leg, if you remember. So it was clogged somewhere, so all that fluid had nowhere to go. So it just kept accumulating in the tissue. So that's just refreshing to remember from before. As far as immunity and non-specific defenses, the lymphatic system is part of your non-specific defenses and specific as well. We're not going to get too deeply into it at all. But basically, if microbes get into your lymphatic system, typically they're screened out. They're filtered out. And one way of doing that is in the lymph nodes. You've all probably heard of lymph nodes before. You might have had swollen lymph nodes like in your neck. There are concentrations of lymph nodes in different areas of your body. You have some in your neck, in your underarm, your groin, different places. See lymph nodes in there, underarm area. And if microbes are moving through the lymphatic vessels, all the fluid that's moving through the lymphatic vessels goes through lymph nodes at some point or another. The lymph nodes are basically filters. And they have a lot of immune cells in them, so they filter out microbes, essentially, and kill them. They filter out microbes, kill microbes, and they're also helping to boost the immune system by doing that. And we're not going to get too much into it. It's deep, complex, exactly what they do. Zooming in on it, you have blood vessels leaking out fluid, goes into the lymph vessels like that. Microbes that get in, they'll travel through, but then they'll hopefully get stopped in here. If you get a swollen lymph node, that's because there's a lot going on there. There's a lot of inflammation going on. A lot of immune cells are trying to fight off an infection. Sometimes it doesn't go down. I had an infection when I was a kid, and this lymph node got huge, and it went down a lot, but not completely. It's this one's a little bit larger than that one. Right? I can't feel this one over here, but I can still feel this one a little bit. Probably some scarring, because I told you inflammation, it causes damage. So I probably had a little bit of damage in there, some scarring in the lymph node. So that's how that works. Phagocytosis, the eating of microbes. Any questions about lymphatic system, the lymph node? And there's other aspects. The spleen is also involved. We're not getting into the details of how the lymphatic system is involved in immunity. There's a lot more involved than what I'm talking. This is microbiology class, not immunology. So we're not going to get too deep into it. Phagocytosis. This is a phagocytic cell. There's different types of phagocytic cells. I mentioned macrophages and eosinophils a little bit. So this is a generic phagocytic cell, and this is how phagocytosis works. You got a bacterial cell, for example, that gets engulfed. This is a cross section, so you're seeing inside. And then there's this. This bacterium is enclosed in this membranous sac. It's inside of there. And then lysosomes come up. If you had Biology 1, BSc 1010, you might remember that term, one of the organelles, a lysosome, which carries lots of digestive enzymes. And these enzymes are going to be able to break down all the macromolecules, lipids, nucleic acids, proteins, carbohydrates, everything that a cell is made of. So it's just it's shock and awe of the bacterial cell, breaking it down. And then you can see it's been broken down here. It's beginning to be broken down more and more. And a lot of that stuff gets excreted or secreted from the cell. And a lot of this will act as the chemotactic agents, by the way. Right? So if this is eating bacteria, it's releasing this stuff, and then another cell recognizes it. There's going to be more and more of this debris that other cells can come in and fight off the bacteria. By the way, some bacteria can survive this, and they actually will grow inside. They might prevent the lysosome from fusing. Bacteria have all kinds of different ways of getting around your defenses. That's one way that will prevent the lysosome from fusing, so it will grow inside this. It can divide. Okay, inflammation. A big part of non-specific defenses, inflammation. It's double-edged sword, inflammation. It's caused by immune system participants. It's basically, inflammation is a way of bringing together all of the immune system participants, immune cells and other things, to fight off an infection. That's inflammation. But there's damage that can come out of that. If it's prolonged inflammation, it can cause damage. My lymph node. Arthritis in the joints is constant, attacking of the joints by immune system cells, to the point where, fingers get mangled. Can't use them. It's so severe. That's rheumatoid arthritis. There's osteoarthritis. Vasodilation, I mentioned that term. So vasodilation is the dilation of blood vessels. opening up. Vasodilation. It's the dilation or the opening up of blood vessels. That's a big part of inflammation. I'll talk more about that in a bit. Margination and emigration of white blood cells, WBC, white blood cells. Margination means, let me go to the picture of it, actually. See if I had a little bit. Right here. Margination is when the phagocytic cells will stick to the inside of blood vessels. So certain immune cells stick to the inside of blood vessels. That's margination. Phagocytes, most typically, stick to the inside of blood vessels, near where there's an infection, basically. Margination. Emigration. Your book uses a different term, diapedesis. I'm going to use the term emigration. But diapedesis is the same thing. Diapedesis. Did I spell that right? Something like that. That's emigration. So I'm going to use the term emigration. Okay? Emigration is when the cells move out of the blood vessels into the tissue. Like immigration and emigration. Immigration, people moving into the country. Emigration, people moving out of the country. Emigration, moving out of the blood vessel. I'm sorry? Into the tissue. Yeah, it moves completely out of this vessel into the tissue. And then you have acute phase proteins activated. These are lots and lots of different types of proteins that are, we call complement, cytokines, and vasodilators. These are all acute phase proteins. There's dozens and dozens of them collectively. There's probably 50 different complement proteins and 50 different cytokines. Not that many vasodilators, but several. So these are lots of different proteins, all involved in inflammation. And I will touch on them again. Actually, I think I already defined this previously. Cytokine is an immune cell signaling molecule. Cytokines are immune cell signaling molecules. Complement, I'll talk in detail about that in a little bit. I won't get to it this time, but towards the end of the chapter, I'll talk about complement. Okay, so inflammation is bringing all the immune system participants, different cells, these things, these proteins. Blood vessels are dilating. The white blood cells are, getting to the tissue where the bacteria are. It's bringing everything together. And you have symptoms, signs and symptoms from that: inflammation, redness, pain, heat, swelling, edema. Because everything's coming into a localized area. You have the redness, the blood leaking out. You can see that. The pain, the fluid buildup, stimulates nerve endings. Heat, because there's a lot of cells there metabolizing. There's going to be heat, swelling, the fluid leaking out of the blood into the tissue. That's swelling. And of course, you'll have more of these things the more inflammation there is. This table here, all I want you to know is the names of these compounds and know that they all can act as vasodilators. They can do other things, cross that out. I just want you to know that they can all act as vasodilators. And they're released by damaged cells. Vasodilation, of course, again, dilation of the blood vessels. So I told you the blood leaks fluid naturally, normally. Well, if you have an infection in some tissue, you want to dilate those blood vessels. And it gets those immune cells out of the blood vessels more easily, basically. That's partly why vasodilation occurs. And I'll pick this up on Monday. See you then.